just a dog in the window. Na 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 na. Wait, is that a river I'm hearing now? It's really a river. I didn't know there was a river in here. I wanna go fishing. Wanna go with me, dog? I got something! There! Now that we have a fish, we can now go home, dog. Let's go! Alas, we're home. Hey, I've been thinking. For some time now, I really wanted to eat curry with raw fish. Is that weird? And now we're going to eat. Here's the one for you, dog. Wait, what was that? I think it came from this fish. Oh my God! No! Oh my God, dog! Crap! We're doomed! We're doomed! Dog, come on, dog, come play with me. Dog, why are you responding to me? Don't you want to play? What's wrong with you? Are you sick? Are you alright? My hello, good morning. How can I help you? Doctor, you have to help me. My pet here, well, his name is Dog. Well, it's as if I think he's sick. I mean, he's always lying around for a couple of days now. Oh my! What could have happened to him? Actually, Doc, I really don't know. Well, for starters, could he have eaten something bad? Like a couple of weeks ago or yesterday? I remember now. I may have had accidentally fed my dog fish with worms. What? A month more or less probably. We're at this river and we were fishing. Then we caught a fish. Then we used that fish for dinner. However, it was only when I have bite that I noticed that it's got some things in there. Did you notice if there were snails in there? Now that you think about it, there were snails in there. Why? Well, Let me explain something to you together with my colleagues. Come on.
Chlonorchiasis is a disease caused by a type of liver fluke called Chlonorchiasis, or otherwise known as Chinese or Oriental liver fluke. It can be found mainly in Eastern and Southeastern Asia, specifically in countries including China, Korea, Vietnam, Taiwan, and Japan. It has also been reported in non-endemic areas, including the United States, where it occurs in Asian immigrants. The worldwide burden of chlonorchiasis was estimated at 19 million persons in 1946 and 7 million in 1994. As of 2011, an estimated 200 million persons are at risk and 15 to 20 million infected. Infection occurs through ingestion of raw or undercooked fluke-infested freshwater fish. Moreover, it is a common infection of dogs and other fish-eating carnivores. However, humans may also substitute as reservoir host. Mainly, it affects body tissues such as the liver, gallbladder, and the bile duct.
Part of the Chlonorchiasis life cycle takes place in an aquatic habitat. The first intermediate host is always a snail of some sort, mainly Parasitology moncoricus, and species from the genus Bulinus. However, since Chlonorchiasis is mainly in specific regions of the world, certain kinds of freshwater fishes are normally found to be second intermediate hosts. There are 12 species of fish that are mainly responsible for passing the infection to humans. Some of these fishes include Pseudorasbora parva in the Japanese region and Tenopharyngodon edelia in the Canton region. Because the definitive host can be any kind of fish-eating vertebrate, its life cycle is no longer confined to an aquatic environment, and its geographic range has the potential to substantially increase. Possible hosts include humans, reptiles, birds, pigs, dogs, and cats.
Chlonorchiasis egg is oval-shaped, 26 to 30 micrometers in length, and has a thick yellow-brown shell surrounding it. The C. sinensis egg has an operculum covering the anterior end, with a conspicuous rim that protrudes from the sides of the egg. On the posterior side, each egg has either a small knob or a little curved spine. Its myrocidium larval stage is ciliated and slightly oval in shape. It has two simple eye spots and lateral papillae, which protrude outwards and serve as sensory organs. The sporocyst resembles a hollow and simple sac. Oftentimes, the developing radiae are visible inside the sac. In the redia stage, it retains a very simple worm structure. In some ways, it still resembles a sac. It has a pharynx, but no esophagus or intestine. Developing cercariae are visible in the rest of its body. The C. sinensis cercaria resembles a small adult with a tail, which it loses upon penetration of the second intermediate host. The tail has dorsal and ventral fins on it to aid in locomotion. It is brownish in color, and unlike an adult, it has two eye spots, penetration glands, and a stylet at its anterior end and a cuticle with small spines. In the C. sinensis metacercaria form, C. sinensis is encysted and does not look like a fluke. It has lost larval organs such as the eye spots, the stylet, and the tail. The round cyst has very thick walls, and within it, the maturing fluke is visible, as are its suckers. C. sinensis as an adult is a very narrow fluke, 10 to 25 millimeters in length, flattened dorso-ventrally, with trematode characteristics such as an oral or anterior sucker, a ventral sucker known as acetabulum, no blood circulatory system, and no body cavity. The common genital pore is just anterior to the acetabulum. The fluke is tapered at the anterior end and rounded at the posterior end. The intestine is bifurcated and ends blindly. A thick and elastic cuticle lacks any kind of spines or scales and can either be a translucent gray color or yellow color due to the absorption of bile. When stained on a slide, its branched testes, lobed ovary, and follicular vitellaria are apparent, as is the long and convoluted uterus. The pharynx and esophagus are also visible.
The life cycle of Chlonorchiasis consists of a three-host system. A snail is always the first intermediate host, followed by a freshwater fish, and finally a wide range of other definitive hosts. Chlonorchis sinensis is hermaphroditic, therefore, every single sexually mature fluke will produce eggs. Each adult yields a daily production of 4,000 eggs for at least 6 months, which are produced by cross-fertilization. The eggs are passed out with the feces of the definitive host. An egg will not hatch until it is ingested by a proper first intermediate host. Once ingested and in the alimentary canal, a myricidium emerges from the egg. It remains in this stage for only about 4 hours, after which it becomes a sporocyst.
The sporocyst is normally located in the wall of the first intermediate host's intestine. It can be found in other organs as well. In the next 20 days, germinal cells in the sporocyst undergo a sexual reproduction to produce a new generation. Each new organism is a rhizia and they emerge when they are mature. Each rhizia contains germinal cells which will undergo further asexual reproduction. The next generation are cercariae. It travels to the hepatopancreas of the snail while the cercariae are developing within itself. At some point, though it is not yet entirely known, up to 50 cercariae emerge from the rhizia's birth pore, still immature. The cercaria leaves its snail host 1 month after the initial infection of the snail by the eggs. It emerges due to some sort of stimuli, although the specific stimulus is unknown. Upon contact with the second intermediate host, it attaches with its oral sucker and penetrates the host. It loses its tail during the process of penetration. After 35 days of infection, the cercaria encyst under the scale or muscle. The metacercaria remains encysted in various tissues until the second intermediate host is ingested by a vertebrate. Excission takes place in the duodenum and a juvenile adult fluke emerges. A juvenile adult reaches the bile ducts within 4 to 7 hours after ingestion of the intermediate host. From the duodenum, it penetrates the gut wall and is carried to the biliary system via the ampulla of Vader. After 1 month, it matures enough to produce eggs.
In humans, acute chlonorchiasis may be asymptomatic or scarcely symptomatic in light infections. But if the number of worms is significant, fever and right upper quadrant pain are not infrequent and may be associated with intermittent colic pain caused by the worms obstructing the gallbladder. Chronic chlonorchiasis, which results from protracted episodes of reinfection over time, may be most severe. The presence of the worms in the walls of the smaller bile ducts causes chronic inflammation, resulting in fibrosis of the ducts and destruction of the adjacent liver parenchyma. Cholangiohepatitis or recurrent pyogenic cholangitis is also frequent. Chronic chlonorchiasis is strongly associated with cholangiocarcinoma, a severe and often fatal form of bile duct cancer. The International Agency for Research in Cancer classifies C. sinensis as a Group 1 agent, which means it is carcinogenic to humans.
I will prescribe praziquantel to your dog as it is the only drug recommended by WHO for treating chlonorchiasis. 80 mg of this should be enough.